everyone! Today, as we bring for you the 12th episode of the Immunology for Everyone podcasts by SKA, I say we celebrate a small milestone. The journey has been both exciting and enriching for all. The format of delivering science, especially immunology, through these podcasts is not only challenging but has brought out explanations and presentations in patterns that will help our audience grasp the very basics, link them to their medical applications in their work and practice. Marking a little closure to the basic immunology concepts after this podcast on thymus independent antigens, SKA can now open the world of application based immunology for you all. I can vouch it is going to be your everyday listen as you set pace in labs each day. So stay tuned for all those little notes that you won't make anywhere else and get your peers on board too. Do share your views, suggestions, and queries with us. Mic over to SKA now.
Hello, my friends. I'm sure you all are doing great and keep listening to this podcast on immunology for everyone. You see that in last couple of episodes, while discussing the activation of B cells, I discussed the interesting phenomena of linked recognition and affinity maturation. These phenomena includes the somatic hypermutation and antibody class switching, which are the hallmarks of B cell activation in thymus dependent antigens. But I think you all know about the athymic mice, which is also called the athymic nude mice, where the thymus is missing in this phenotype. We do have a similar situation in humans also, known as congenital athymia, which is an ultra rare disease characterized by the absence of functioning thymus. So, in these humans and mice, there are no T cell development. But in spite of having no T cell help in these humans and mice, antibodies to many bacterial antigens are still detectable. So, this is because of special properties of some bacterial antigens, mainly which are the cell wall coatings of some bacteria like polysaccharides, polymeric proteins, and lipopolysaccharides, which enable them to stimulate the naive B cells in the absence of even helper T cells. So now let's discuss the thymus independent antigens. These antigens are known as the thymus independent antigens because they stimulate strong antibody responses in athymic individuals also. These are generally non-protein bacterial products which cannot elicit classical T cell responses, yet they induce antibody responses in normal individuals. However, it has been seen that B cell responses to these thymus independent antigens are also influenced by the presence of T cells, maybe indirectly through cytokines such as interleukin 5, and the B cell responses are greatly diminished in these animals that have no T cell at all. So, such thymus independent antigens they fall into two classes that activate the B cells by two different mechanisms. First of these kinds are the thymus independent type 1 antigens, which possess an intrinsic property that can directly induce the B cell division. So, at a very high concentration, these molecules cause the proliferation and differentiation of most B cells, regardless of their antigenic specificity. And this is known as polyclonal B cell activation, which you might have known that some of the bacteria and parasites, like Leishmania parasite, it induces polyclonal B cell activation, which leads to generation of lot of immunoglobulins, which include some non-specific but some antigen-specific antibodies also. Such antigens are known as B cell mitogens because they induce mitosis. So, a mitogen is a substance that induces cell to undergo mitosis, which is the cell division. So, for example, we have bacterial lipopolysaccharide, which binds to lipopolysaccharide binding protein, and you know that it also binds to the toll-like receptor 4 on B cells. But B cells, they require about 100 times higher concentration of lipopolysaccharide than those which are required for to activate the dendritic cells, because dendritic cells and macrophages, they all express TLR4. Uh, so B cells can get activated by these, bacterial lipopolysaccharides. But these thymus dependent type 1 antigens, they are insufficient inducers of affinity maturation and uh, and also the memory B cell production, which also require the T cell help. Still, such responses have an important, clinical role in the defense against several extracellular pathogens, because they induce a very quick antibody response. Then we have the second class of thymus independent antigens, which consist of molecules such as bacterial capsular polysaccharides that have highly repetitive structures. These thymus independent antigens are known as thymus independent type 2 antigens, and these antigens do not contain any intrinsic B cell stimulating activity. So they are categorized as type 2 antigens. So these antigens can activate only mature B cells, because immature B cells would get inactivated by repetitive, epitopes. This may be the reason why infants and children, they do not make antibodies to polysaccharide antigens efficiently, because most of their B cells are immature. These antigens most probably act by extensively cross-linking the B cell epitopes receptors of mature B cells specific for the antigen. So all mature B cells, they express many B cell receptors, and these polymeric antigens, they cross-link these, and they can allow the activation of these mature B cells. However, it has been seen that excessive receptor cross-linking many times renders the mature B cells as unresponsive or anergic, just as it also does for the immature B cells. And so, the epitope density seems to be crucial in the activation of B cells by uh, thymus independent type 2 antigens, because at very low concentration, the receptor cross-linking is insufficient to activate the cell, and with a very high level of density, the B cell becomes energetic. But you know that while these type 2 antigens alone induce a production of IgM type of antibody by activated B cells, because that is the primary response, as we all know, is the IgM type antibody. So the activated, sometimes the dendritic cells, they get activated also, and in response to some of these bacterial antigens, the dendritic cells, they release a cytokine, which is called the B cell activating factor, also known as BAFF, which augment the production of antibodies against these bacterial antigens, and it also induces class switching. So that is a very interesting phenomena, that even the thymus independent type 2 antigens, they can induce uh, class switching because with the help of dendritic cells which produce uh, B cell activating factor. So this B cell activating factor is a member of tumor necrosis factor family and is fundamental survival factor for transitional and mature B cells. So the overexpression of this particular cytokine can lead to an expanded B cell compartment and can cause autoimmunity in the mice. And it has been seen that even in humans, elevated levels of this BAF can be found in the serum of many autoimmune patients. This cytokine and its receptors, they have a role in B cell biology outside of the survival mechanism. So this include the germinal center maintenance, isotype switching, and regulation of specific B cell receptors. So, whenever this cytokine is there, there is a lot of B cell activation, there is germinal center formation, as well as memory B cell formation. So, the B cell responses in thymus independent type 2 antigens provide a prompt and specific response to an important class of pathogen, which are basically extracellular bacteria. And the examples of extracellular bacteria are Staphylococcus aureus, Staphylococcus pyrogenes, Pseudomonas aeruginosa, Escherichia coli, because these are classified as uh, extracellular bacteria. And because many of these common extracellular bacteria pathogens are surrounded by polysaccharide capsule, that enables them to resist the ingestion of by the phagocytes. So these bacteria do not only escape direct killing by phagocytes, but also avoid stimulating the T cell responses. Uh, Although the presentation of bacterial peptides by macrophages, they can sometimes induce some level of T cell activation. And the antibody that is produced rapidly in the response to these polysaccharide capsule without the help of peptide specific T cells can coat the bacteria. So these antibodies, they help in um, ingestion of these bacteria by opsonization. In this context, we already discussed the importance of antibodies to the capsular polysaccharide of Haemophilus influenzae type B bacteria, which we discussed earlier. The Hib vaccine, you know. Um, so the which is thymus independent type 2 antigen, and it induces protective immunity in the to this bacterium, while the adults are capable of making antibodies to this capsular polysaccharide, whereas in the children you have to have a conjugated vaccine. And I told you that the polysaccharides are conjugated with the tetanus toxoid, and the T cell responses against tetanus toxoid provide the T cell help for the production of antibodies against this polysaccharide capsule in the children. So that's a very interesting phenomena. So it is clear now that the T dependent humoral responses, they generate long lived memory cells and plasma cells, which are predominantly through the germinal center reaction. But it is interesting to find that in humans as well as in mouse, memory B cells and long-lived plasma cells are generated, including bacterial polysaccharides. Although the underlying mechanisms for such T cell independent humoral memory are not very clear till um, at the moment, But very recently, it has been reported in a seminal study by Xin Liu et al., which is published in Journal of Experimental Medicine in 2022, that the T independent antigens can induce germinal center formation, uh, but these germinal centers are transient and thought to be non productive. By these authors, by ingenious genetic fate mapping studies, they found that these germinal centers actually uh, output the memory B cells as well as the as well as the plasma cells, which make long lasting antibodies. So using what they did was they used a conditional BCL6 deletion approach, and they showed that memory B cells and plasma cells, they fail to last when the T independent germinal centers are precluded, suggesting that the germinal center experience per se is very important for programming the longevity of T independent memory cells and the plasma cells. So, consistent with the fact that infants cannot mount long lived humoral immunity to T independent antigens, it has been shown that the B cells from young animals intrinsically fail to form T independent germinal centers. So the results from this study, they suggest that T independent germinal center support humoral memory and germinal center induction may be the key to effective vaccines with T independent antigens. So, we always understand that germinal center support is very important even in T independent antigens for formation of the long lasting immunity. So, guys, with that, we have covered the activation of immune response to many pathogens. Uh, I would like to close here with the hope that many young brains will get stimulated to take up some of the research questions that we have discussed uh, in these podcasts, where we discuss the activation of T cells and activation of antigen specific B cells. Now, you guys may go back and listen to any of these episodes, as these are available on YouTube channel SKA Talks, and you can revisit all those memories. Further, I plan to take up some basic techniques as we use in the immunology laboratory for diagnostic purposes or for research labs. And I plan to take up disease specific immunology like cancer immunology, uh, vaccines, and all. You may also send your suggestions in the comment box for any specific topics that may be of interest to you guys, and I will see that if I can cover some of these in the subsequent episodes. So, guys, keep listening and do read some of the material as quoted here, with a hope to bring another interesting aspect of amazing immunology next time. Goodbye for now, and thank you very much.